Be live, sir.
Okay.
Started.
Good evening, ladies and gentlemen. Future scoping is the theme on menu today. INSA has taken several initiatives during this pandemic period, and this web discussion is just one of them. We have the chair, Professor Chandrima Shaha. Professor Shaha is president of Indian National Science Academy, New Delhi, and also the J.C. Bose Chair, Distinguished Professor, National Academy of Sciences, Indian Institute of Chemical Biology, Kolkata. Earlier, she had been the director of the National Institute of Immunology in New Delhi and also emeritus professor. She has been an integral part of the Indian science fabric and needs no introduction to this audience. It suffices to say that she's a leading life scientist of the country and well recognized all over the world. She's fellow of all the major academies of India and also TWAF and has been conferred with a number of awards and medals. I request Professor Shaha to please welcome our guests. Professor Shaha.
Members of the distinguished panel, distinguished fellows and friends, it's a great pleasure to welcome you all today to this exciting program under the INSA SERB, which is Indian National Science Academy and Science Engineering Research Board Future Scoping Initiative. INSA co-hosts this program with SERB, with whom we have entered into a very fruitful and productive partnership. The idea is to trigger discussions on contemporary subjects of relevance, particularly those that may strongly impact science and society. Gene editing is one such frontier area that has caught attention on account of immense opportunities hidden in it. Be it for plants or for animals, gene editing will revolutionize the world in not only health, but also the food scenario, one of the major concerns of the world today. In fact, by providing us power to modify the plant genes, it even promises to influence the environment positively, as we shall be empowered to develop novel, efficient varieties of crops. There will be a lot of scientific, technical, ethical, legal, and other issues to be sorted out, and we are continuing to work on these issues through these kind of discussions. We have with us Dr. Rakesh Mishra, Director of Tata Institute of Genetics and Society, as convener, with veteran Professor Deepak Pental, former Vice-Chancellor of Delhi University, along with Dr. Amitabh Mohanty and Dr. Debojyoti Chakraborty. Dr. Mohanty is currently the Global Biotechnology Operation Leader at Corteva Agriscience, USA, while Debojyoti Chakraborty is a Senior Scientist at IGID. Dr. Mishra is well known, but I shall still say a few words about him. After PhD from University of Allahabad, he did his postdoctoral research at the Indian Institute of Science, University of Bordeaux, France, St. Louis University, the University of Geneva. He then joined the Center for Cellular and Molecular Biology, Hyderabad, and became its director in 2016. His research interests are comparative genomics of non-coding DNA in the context of evolution of complexity, genetic basis of anterior-posterior body axis formation in animals, and role of epigenetic regulation in development. The Atal Incubation Center at CCMB played a key role in genomic surveillance of the SARS-CoV-2 virus in India during the COVID-19 pandemic, and it is a testimony to his leadership. Dr. Mishra is an elected fellow of the major science academies in India and is recipient of several honors and awards, including the J.C. Bose National Fellowship. So I welcome you all to this discussion and look forward to your active participation. Please be on the lookout for more activities of INSA on Facebook, Twitter, and website. For example, we have 2 days of conference on 23rd and 28th by our Kolkata chapter. Please attend those, and wish you a very intellectual entertainment for the next hour and a half or so. Thank you very much. Over to you, Dr. Mishra.
Thank you. Thank you, Dr. Chandrima Shaha, for the very generous introduction. And thank you really for initiating this activity in coordination with the and I think this is the responsibility our academies uh, have to take care when the new technologies are emerging. Scientific leadership has to be brought on board, and so that the people and policymakers are communicated what is coming in the near future. And the future scoping is something extremely important. I'm honored to be coordinating this session with very distinguished speakers. I mean, all the three speakers are really in the thick of this emerging technology and have contributed immensely in a very visible, with very visible impact. So, so before I invite the speakers, the format we are keeping is that I will have a few minutes of introduction of the this topic, and then I will invite one after other the three speakers, and at the end of the all the three talks, then we will have a joint question answer and discussion session. And I request all the viewers to put their questions in the chat box, or later they can use the uh, reaction button and ask questions once we are done with the presentations. So I will just share the my very short presentation. Uh, as I mentioned, this is a really a very, very timely and important initiative taken by INSA and SIR under the future scoping initiative about gene editing. Uh, so before we go to gene editing, the genome revolution really played the key role in setting the platform for this technology. Genome sequencing, as we see today in terms of time, it's not years or months. It just takes days to complete the genome sequence. It doesn't cost millions of dollars as it used to be a couple of decades back. It actually costs hundreds of dollars and is going to be lesser and lesser. And it's not the scale that few genomes are sequenced. Now hundreds of thousands of genomes are sequenced, including humans and complex organisms. So that's the revolution that has taken place. Advantage of this is that we can have deeper understanding of the biology beyond the model system, because we can directly ask questions on the organism of choice and go and, as you will see later, modify the genomes and ask questions about that part of the genome. We can look at the genome at the population level, which allows us to look for personalized and precision medicine. What is more is that genome editing brings really the ultimate tool of genetics and genomics, where we can not only read the genome, we can go and edit as and when we want. This opens, as Dr. Saha already mentioned, unprecedented opportunity in agriculture and healthcare, and our speakers are going to cover those issues in certain details. So I will just take 2 minutes just to simplify what is genome editing. Genome editing is very simply create a double-strand break at a desired site in an organism or in a cell, and repair it with intended template, by which you also slip in your envelope of choice or wish that you want to introduce at that point in the genome. And there are multiple ways of doing it. So there are the zinc finger nuclease or TALEN approach or CRISPR-Cas9 approach. All these approaches essentially are making a break at a particular point in the genome in a very specific manner. And once that double strand break is created, then you provide the template to change the process, change the genome at the exact site in the process of repair. So in very simple terms, you cut, this decides where you are going to include the change, and then you introduce repair which means what you are going to do. And then very simply, you convert one part of the genome into a desired part as you want. So this really simplified uh, uh, approach, and lots of labs are using in the country genome editing for various experimental systems. In terms of future scoping, we are using this in the lab, but this technology is actually very much poised to deliver really in a major way. So for example, we can introduce new genes in an organism and produce pharmaceuticals. That's major biotechnological applications of this. It is going to be crucial in crop improvement, where we will reach a position that we don't need pesticides, we will have more nutrition, we will need less resources like water for growing the crops. We can have tailor-made model organisms to study biology, to understand the human disease in using the model system. We can have livestock improvement for more productivity and less impact on environment. And we can correct genetic disorders, particularly somatic changes, and germline changes are not allowed in most places. And then there is another interesting aspect of gene drive, but the environmental impact and ecological impact of those needs to be looked into carefully before those things are introduced uh, into the and in the real ground. So these are most of these things have certain stages of approvals, of trials, and so on. And these are the issues that our experts will discuss. So what we need to do at the moment is we need societal outreach, and it is the responsibility of this community to convey the message of the technology and build opinion and bring in acceptance from the societal of ethical and safety concerns. And when the technologies of higher impact, we have to be very careful that we are adopting safe and ethically correct policies and guidelines. So it's clear that technology has arrived, and we should adopt and benefit from it. And that's the purpose of this, one of the purpose, a small step of this event. And I hope that with the presentations that are going to follow and the discussion that will be after the presentation, we will have interesting outcomes, and which will be going long way in using these technologies for societal use one day. With that, now I would like to, okay, I have to stop sharing. Right. Yeah. So with that small introduction, then I would like to invite first Professor Deepak Pental, the former Vice-Chancellor of the University of Delhi, to make the first presentation. As Dr. Saha already introduced, Professor Pental has been in the really involved in this kind of technology, and he has been leading from the front. His work is known, and his progressive approach for the agriculture is very well known. And he's going to discuss about how the technology of genome engineering, genome editing is going to be important for from various aspects, particularly from the aspect of small farmers. In our country, large number of stress is with the small farmer, because most of the farmers are a small holding, and technology should be developed keeping those interests in mind, because that will have a larger societal impact. And I would request Dr. Pental to give in the beginning. Please put your questions in the chat box, and we will discuss at the end of this set of presentation. So, Dr. Pental, Can you share your intention, Esther?
Thank you. It's a pleasure to be part of the panel and to speak on the subject. I will try to put into perspective of why do we require these new technologies, particularly gene editing. Dr. Mishra has already given the background, and I'm sure anybody who is reading anything on biology must publications, virtual explosion in publications on gene editing. There has been tremendous work on finding new enzymes after the paper by Doudna and Charpentier in Science in 2012. Engineered proteins, base editing, prime editing. I mean, it's difficult to cope with the developments in the project in this area. And I think the only area which beats this gene editing area is COVID research, because that has affected us very badly, and the whole global level, the concern, there is a deep interest into COVID related research. But after we leave that COVID area, the maximum number of publications you will find in the gene editing area. And we have to see why we are where we are, why we are going so slow while the world has moved so fast. And I'll try to put it in a perspective of agriculture. Why do we require this technology rather desperately? The plant breeders have been irradiating plants for so many years now, trying to get some useful mutations. And as Dr. Mishra pointed out, with so much knowledge on genomics today, almost all the crop plants that are grown on the face of this planet have been sequenced, not only crop genomes, but the germplasm, the wild resources. The huge data has been developed on the genomic side. And as I was mentioning, there are tremendous improvements and developments in terms of gene editing techniques itself, providing unprecedented opportunity to deal with plant genomes. And I would say something about insect genomes also, because the biggest threat to plants comes from the insect pests. So if I can start with my slides. The big goal in agriculture is that we should have low input and high output. High output is required because population is increasing in India. Still, it will plateau out at about 1.6 billion, which was the entire population of the globe in 1900. So we need to reduce the inputs of natural resources like water, because we are over exploiting our water resources. We have to optimize our fertilizer use. We have to optimize our irrigation. Agrochemicals also cost, and we have to protect the crops. And there is expenditure on tilling the land, on harvesting, and on buying good quality seed. So all these inputs, how to keep them low and how to get higher output so that the farmers can make some money, particularly the small landholders. I'm not going to go into all these aspects. I recently wrote an article in Dialogue, which I have circulated to a number of colleagues who are members of the academies, and I try to reach out to INAS members also. this paper is available on the dialogue site and gives some arguments on the inputs, how they can be optimized and how the output can be increased. Just to remind all the listeners, the global population has really increased in the last century. There were 1.6 billion people in 1900, and India alone is going to have 1.6 billion people when the population plateaus out in 2040, between 2040 and 2050. How have we fed all these people? Before 1900, the major increase in yield basically came from increasing the land under cultivation. The North American continent, the South American continent, Australia, many parts of India which were forested were brought under cultivation. So the main expansion was, the main expanding of food basket was due to increase in the land under till. And since 1900, it's a miracle that we have been able to feed this huge population increase, basically because of nitrogenous fertilizers, the Haber-Bosch process, mechanization, which meant less fodder requirement, crop protection chemicals, and systematic plant breeding. And gene editing and others are the second phase of systematic plant breeding. The first phase was the rediscovery of the Mendelian laws of inheritance and making a proper statistical framework for doing plant breeding in terms of multi-site trials and so on and so forth. Since 1960s, those of us, the younger people, would not know these years, but in the 60s, India was staring at a famine-like situation, and we were importing a lot of food grain, and then the dwarf wheat and rice were introduced into the country, and that was the big green revolution. The land under cereals has hardly increased, maybe by 5 to 6%, and the population has gone up by about 150%, but the cereal yield and cereal production has gone more than 200%. That means people are consuming more and people have been able to ward off starvation in different parts of the world. But I want to tell you that this green revolution did not occur only in India. Before that, you will see that the wheat yields in Britain, who the country is good at keeping good records of their wheat yields, and you will see that the big yield increase has come only by the middle of the last century. And if you look at corn, another success story, till 1930s, the corn yields were less than 2 tons in the United States, but today they are touching 10 tons per hectare. So the classical breeding, at least in the cereals, the hybrids and dwarfing genes, the biggest jackpot the humankind has hit in the last century, with which we were able to feed the people and have superior food also in terms of corn being fed to animals and a lot of animal protein also being available for nutrition. Now, I would like to highlight the one aspect which I believe is the most important in maintaining the yields, in managing the climate change, and in giving better deal to the farmers. And that is by protecting the crops, increasing the yield by hybrids and through classical breeding and through marker-assisted breeding now are important areas, because our yields are still quite low as compared to the global standards. But I would like to draw your attention to the fact that like human beings have all kinds of diseases and pathogens attacking them. Same is with the crops. If you take rice, there's the blast disease, sheath blight, bacterial blight. There are insect pests, stem borer and plant hopper. And if the crops are not, if rice is not protected, you will have a famine on hands. Same way with wheat. Usually we think that the green revolution was about halving genes, but we should spare a thought for the fact that huge effort has gone into breeding resistant lines of wheat, which are resistant to rust diseases, smut diseases, and without this resistance conferring genes, the wheat yields will either dramatically come down or we will have to use much more of fungicides than we are currently using. Same way with maize. There's a big attack of the African Spodoptera species, and this fact that the crops are very vulnerable to both to all kind of pests and pathogens which invade them and consume them. You will see here some of our legume crops, which where average yield is only about 1 ton per hectare. We want to replace wheat and rice with these crops, but it's going to be pretty tough because the yields are very low. While wheat and rice have been very intensively researched upon in all over the world, and we benefit from our own generated knowledge and spillover knowledge. In case of these orphan crops like pigeon pea, chickpea, mung bean, even in cotton, the insect pests and pathogens are causing havoc and reducing the yields of these crops. I have been working for almost all my research life in India on a crop, oilseed crop, mustard, and we are currently importing about 70 to 80,000 crore worth of edible oils because most of our oilseed crops are stuck at about 1.1 ton per hectare. And then the number of biotic stresses is very high. I have depicted 5 here. The top column is stem rot, which has become very vicious in the past 5-10 years. Alternaria blight, which is not allowing the crop to be grown in the hilly areas. White rust, for which we discovered some genes and can now be tackled, but it has been shaving off about 10 to 15% of the yield. A root parasite, Orobanche, leaves the plant high and dry. In other countries, as I said, our yields are poor in these crops, even though the rapeseed mustard area is now under protective irrigation. 80 to 85% area has been put under some or the other irrigation, even in dryland areas like Rajasthan. But still we are struggling with yields because of these diseases and the pest problems. So how do we protect our plants and give our farmers a better deal and make sure that the country remains secure in terms of food production? We have new technologies which are all basically have originated from the recombinant DNA revolution. As you know, after the Watson and Crick's paper on DNA structure, and that's the beginning of the big molecular biology area developments, and recombinant DNA has been one of the areas which has emerged from the basic knowledge that we gained in molecular biology. Molecular markers and marker-assisted breeding has become quite prominent. This makes the breeding processes more precise. allows us to deal with quantitative traits. We have made a late beginning in this area, but some work is happening, and I hope that India will be able to do well in terms of using molecular markers and marker-assisted breeding, diversification of traits. There's very good work going on rice and wheat, but in other crops, we could do much better. Genetic engineering, we remained confused about it. It's a very powerful technology to add on add-on functions which are missing in a crop. I still believe that we will have to come around to accept genetic engineering technologies, of course, with the due precaution. And gene editing is a new area which allows us to do site-directed mutagenesis, which allows us to make some mutations that we want to based on our knowledge of the genomics. And as Dr. Mishra pointed out, genomics is the key, so much data, and it will be a tragedy if this data is not used and utilized properly by a country like India, which is so much dependent on in agriculture, health, and all other issues. And the recombinant DNA revolution, we need it very badly, as much as any part of the world. In gene editing, I would flag that gene editing can be very useful for changing the oil and meal composition. It can be useful in so many ways. In fact, there are limitless ways in which, and every other day people are discovering new ways of using it. We have done very little structure-function relationship in plants in India. Mostly the work has been done on model systems like Arabidopsis, but now the crops are becoming model systems, and there are pathogen susceptibility genes which can be removed to give the plant resistance. The nutritional quality can be changed, and even in disease resistance, if we have the knowledge of an allele which gives resistance, we can change the allele in our crop plant which has become susceptible towards the sequence which will provide resistance. The simplest thing is double-stranded break, and which comes as STN1, and that is the simplest technology. The other ones are little complicated. The frequency is much lower when you want to do homology-based sequence changes. But there are so many new developments in the area that like the base editing and prime editing, which have been very well reviewed in a recent review, which our colleagues have written, 3 of them from India, which has been published in Nature Plants recently. So where do we stand? The green countries here have accepted STN1 and STN2. STN1 creating breaks and creating mutations, and which is the simplest form of gene editing, and STN2 where we can change the allelic configuration and make some changes in the nucleotide sequence to see how it affects the function of the gene. The third area is where you want to introduce a trait and new gene into a directed site. I personally don't see any reason why we should direct it to a particular site, but there may be some sophistications involved in that so that the transgenes go only in a particular area of the genome and not all over the place as it happens with the agrobacterium mediated transformation or with gene gun experiments. So if you look at India, we are still dithering on what to do. I must commend the National Agriculture Academy of NAS for deliberating on the issue and making very strong recommendations to the government that SDN1 and SDN2 should not be under the biosafety regimes which we have put on the genetically engineered plants. Countries like Japan, which never accepted genetic engineering, have already released gene edited plants. So we have to make a choice. The only exception is a country which puts a lot of resources into R&D. China has invested so much funding into this area. They have probably the maximum publications in gene editing in plants in some of the top journals of the world. They are leaders in plant genomics, but they have not still made up their biosafety regulations. Quietly and silently, I think they must be doing something, and the world will come to know one of these days. But if you see India, our country, with all the improvements that we require in agriculture, and I also want to say here that we need to tackle the insect pests of crops, sucking pests, and all kind of pests. We must start work on population genetics and genomics of these pests, and we must look at the technologies which are based on gene editing to suppress the population of these insect pests and to make some changes that they are not able to carry the viral diseases which are causing a lot of losses in agriculture. I'll finally end by saying, drawing the attention of all of you to the fact that we are not investing enough in R&D. Even if we have interest in gene, we will have to increase our investments. You will see here the investment these countries are making in R&D. India is around 0.7% of its GDP. The only country which has come down in the recent years is Russia. It is still putting about 1% of its GDP. China has increased their investments much more. And you can see that other countries are also increasing, but these countries are very strong base in R&D already. So I would request the president of INSA to get on with other presidents of other academies of science and technology and engineering to ask the government that in the 75th year of India's independence, we should commit to increasing our investments into R&D at least to 1% of our GDP. And we are far behind in gene editing, genetic engineering. In marker-assisted breeding also, we have to make it a routine affair in our laboratories, and this will require more resources. And if we want to get into genomics of insect pests, and then we have to do a lot of genomics work in the country and a lot of population genetics work. We have to have human resource also, which is competent to deal with all these areas. So I think I've said enough. Thank you.
Thank you. Thank you, Professor Pental, for very interesting and touching on the topics which are of a very, very high importance in the current scenario. And I think all of us agree that if India has to become a developed country, if we have to take care of the healthcare and agriculture issues, we have to invest more in research. This will not be possible by importing technology because others are not going to solve problems which are unique to us. So we will thank you for bringing that up, and I think we all have to work together to convince policymakers and government and to public that science is important. And I think in some aspect, COVID has helped drawn attention towards the science. So maybe we should take advantage of that. So we will come back to these discussions. I request all the participants to use the reactions button or put in the chat box their questions. So we will take them up after the presentations are done. So the next, who is a leader in leader in the global biotechnology operations, Corteva Agrisciences. Dr. Mohanty has been involved in really setting up quality infrastructure for genome editing technology to work in the country, including development of competence such as CRISPR-Cas enabled advanced breedings. And he has been involved in many committees and task forces of the government of India in advising. He played a crucial role in Rice Genome Project, and so I'll request him to tell us about certain aspects of gene editing in agriculture, and also if we can, because that seems to be one of the requirements at the moment. So Dr. Mohanty, please.
Sir, first of all, my sincere gratitude to INSA sir for the invite, and it's a proud privilege for me to share the virtual dais with the luminaries of Indian science. So thank you for the opportunity. And thank you, Dr. Mishra and Professor Pental for the nice background. So I will skip some of the slide, because we want to go to the main content, which was which is something that I want to cover. So in today's presentation, what I want to do is I want to talk about the technology, give some examples, and hopefully I'll give some example which you may not be familiar with. And of course, I'll also give some example which I have been talking for quite some time. I would also like to bring your attention to some of the newer opportunities. Professor Pental made that comment that how the productivity has gone up because of hybridization. And as you know, that Corteva Agriscience, which was known as Pioneer at that time, was the company which started the hybrid corn, way back in 1920s. And since then, we have improved the productivity of this crop tremendously. So some of the other ways of crossbreeding are very important part of getting variation and using that for improvement of varieties or hybrids, mutation breeding, transgenic breeding, and more recently genome editing. Now, if you look into the details, one of the biggest game changer of the genome editing is the time saving, right? If you have the ability to transform into elite lines, it really cuts short your product development time, and hopefully the regulatory aspect as well. So that opens up a lot of opportunities for everyone. I will not dwell on this because Professor Mishra spoke about the various means, and these keep changing because we keep on having newer kind of enzymes with newer functionalities. So let's keep. But this is one of the most important slides that I would like to share. So as spoken by my previous speaker, this genome editing is not just only genome editing. Having a really good high quality reference genome, ability to have the double stranded break by various enzymes, have the ability to have the molecular analysis system to get into the analysis of those lines generated, and of course elite light transmission. All these together is what is required in order to be successful. And we have a range of technologies. For example, we use short read sequencing, long read, as well optical mapping. All these come into play in our genome editing effort, and then hopefully you'll see some of the examples down the line. I would like to again emphasize the ability to transform the elite lines, and some of us who have been doing transformation for a long time, transformation still today is not easy, right? And for many of the crops, it's really challenging. I would point you towards some of the technologies that we have developed. For example, with this morphogenic gene that was published some time back from us, you could reduce really the timeline from 4 to 6 months to 1 to 2 months. Now you do not have to, with this help of these morphogenic genes, you do not have to have go through this callus proliferation and somatic embryo stage. You can directly go from zygotic immature embryo to somatic embryo into the plant. and that plays a big role in the success downstream for the genome editing. Again, I cannot emphasize enough. You will see one example where we are doing large scale changes, and that needs the ability to detect the changes. It could be a small one base pair change, or it could be like tens of megabases. And again, all these technologies, for example, short read as well as long read, SBS. I would again refer you to this particular paper that we published several years back. Which allows us the ability to look for the lines that we are going to move forward with. How do you use CRISPR-Cas? I'll again give you a couple of examples. This is probably you are familiar with this particular example. Vaxi, this was one of the first that we attempted. So Vaxi-Con is commercially available, and also what happens is this particular one. This has got translucent appearance. And it's a combination of 75% amylopectin and 25% amylose. So this is used for animal feed, ethanol production, human consumption, all of these, right? This particular mutation, if you look into, there are about 200 Vaxi mutant alleles. These are spontaneous mutations, and Vaxi C is the most common allele that has been used to have this trait integrated into the commercial lines. Now, as you know, backcross breeding takes a lot of time. It takes 6 to 7 generations to bring this mutant allele to our elite, and still the hybrids yield about 5% less than the counterpart, or no, it's a hybrid. And again, to emphasize that it has been used for this particular allele has been used for human consumption, industrial use for a long time. Let's look at the genome editing, what we have done. So we created a number of different variations, two of them are shown here. For example, we deleted the Vaxi C that we are talking about in the previous slide is a 30 base pair naturally occurring mutation. What we created is two of the forms depicted here. We deleted the entire gene, which is about 4 kb over here. And we also, in another case, we deleted or edited the gene with the promoter. And then what are the results? You can see on the right side, we are able to see the conversion of the amylose to amylopectin. So in this particular edited line, 97% of the starch is amylopectin, right? And that can be confirmed by iodine staining, which is shown. Now let's look at the yield. These are yields from the hybrids, and you can see in many cases, out of in this case 5 of them, you can see on an average 5.5 bushel more in yield across this range of hybrids. So and the editing not only resulted in having the trait of Vaxi mutation in our elite line, it also improved the yield. And as you recall in my previous slide, there is yield drag when you try to bring things from non-elite to elite, and that with genome editing, since you are directly transforming your elite line, you can get rid of that. Let's move to another example, which is northern corn leaf blight, and this is another example where this is a fungal disease caused by the exerohelium, tassicum, and this is this can be potentially really bad. It may account to up to 15% of corn yield due to this disease. Ranging from 50 to 500 million bushels. And I'll show an example where we are able to edit this particular line, this particular gene, and we are able to bring in these NLB resistant allele from another corn line to the target species. And the picture here shows you the symptom after inoculation. The left one is a hemizygous plant, and the middle line is a null, and right is a homozygous. And you can see that there is no disease symptom in the homozygous line. So we are able to bring in this resistant allele at the endogenous locus after deletion of the susceptible allele. So all these are possible. The next example is something is quite exciting to me. With and this is a combination of a number of different expertise. And here is an example how genomics can play. This particular example I'm showing in a project trying to do pan-genome of a large number of elite lines for us. We found that there are out of 66, 9 of these line. We found, during the pangenome assembly, we found that there are large scale deletion, insertion, as well as rearrangements, and one of the rearrangement is like 26.7 MB inversion. So this is we found that 9 out of 66 line is showing this, and there was in another 3 line there are bigger pericentromic inversion, which is 75.5 MB. And these inversions are interesting because what happens is these doesn't allow the homologous combinations, homologous chromosomes to pair and recombine, right? That means it excludes a particular region of the genome from recombination. And idea is that we want to have more recombination so that you can select for favorable allele, and this kind of inversion doesn't allow. On the other side, flip side of it, when you have assembled your trait in a complex trait loci or a QTL or a favorable allele, you probably do not want to have this recombine further, so that you can carry forward, and that is also a benefit. So what we try to do is, my colleagues in Corteva, like they try to see, can we, flip this back, this 75.5 MB, can we flip it back to original situation with genome editing? And that's what is schematically depicted here. You can see how pericentric inversion can happen. If you have double-stranded break across this centromere, it can lead to non-homologous end joining and finally results in chromosome with intrachromosomal translocation or a pericentric inversion. So what we did, we selected 6 guide, 3 on each side of the break in this case centromere, and then tested them in transient system about their efficacy, and 2 of those were found to be highly efficacious, and that was carried forward. So we are able to generate 3 plant out of 1500 that we found has got let me spend a minute or so to explain this schema. So we are able to generate 3 plant that we are able to confirm with PCR that this does have the pericentric inversion, flipped back to the original situation. So if you look into the top panel left side, if these primer combination F1 R1 would able to amplify the junction, and F2 R2 in the normal situation, which is the wild type, right? If there is an inversion, so you would be able to see these with a similar kind of situation, but with a different set of primer F1 F2 and R1 R2. So what we observed like that in both plant 1 and plant 2, we are able to see that that inversion has happened, because we are able to get with this particular set of primer F1 F2 and R1 R2 2 of the expected band that we knew that would amplify. In a third plant, we found only one side of the joining has happened, not on the other side, so that's something different. And this was again confirmed with bionono mapping as well as junction sequencing. And that's why genomics come to big play. You need to have all these capabilities, otherwise there is no way that you would be able to confirm all these. And we were able to do this confirmation first with PCR, second with bionono mapping, as well as subsequent junction sequencing, and we were able to confirm reversion back of this particular inversion. And this has key implications for our bidding. So let me spend a couple of minutes for something like a new forum for us that we call it Open Innovation, and this is a forum where we try to aspire to collaborate with thought leaders and innovators around the world to access and develop most innovative technologies and rapidly deploy these technologies through organization and good stewards for those techniques. And there are a number of different areas that we focus on, starting from trade, plant breeding, enabling technologies, biologicals, digital, as well as crop protection. And I would request you to visit this site. There are open grants that are invited, and we do get a lot of requests for this. Let me share also the collaboration that we have in this space, which are listed in the next slide. So right now, some of these examples are listed over here. For example, with CIMIT, we are working on maize lethal necrosis disease. We are making great progress with ICRISAT. We have a couple of crops in which we are working again in genome editing, making good progress over there. With JS Simplot, the collaboration is bruising and browning resistant potatoes. There is a cassava improvement with Danforth Center, with Embrapa Brazil, farming competitiveness and sustainability, and with Zikal photosynthetic capacity and efficiency. Let me talk about a couple of slides about the advantage of CRISPR. Why this is such a great technology, and as one of the first initial practitioners, so this is great technology because it's a relatively simple and very efficient technology. It's a precision editing tool, allows one to change the DNA in a targeted manner. It's quite cheaper and faster than transgenic technology. Rapid product development cycles I showed you earlier, it can reduce by half. And if you are talking inbreds, probably even less than that. Hopefully, with lower regulatory cost and time, would allow smaller players also living playing field, and that's something that we see happening. What India needs as a country, India needs the strategy to have a comprehensive science-based risk proportionate regulatory policy. We have excellent molecular biology and genomics lab, but we still continue to develop capacity and competency from the genome editing areas. Consideration of technology access and freedom to operate is also critical, and I think we need to pay some attention over there. And with that, I'll move to the couple of regulatory aspects. So one thing that I want to highlight here is that gene-edited plants are indistinguishable from the plant that could be produced with conventional breeding, and which have a long and safe history of use, And it is the characteristic of plant and not the production method that determines the safety. It is important that we have a transparent, consistently applied, enforced, and globally harmonized regulatory policy, and we need to have science-based, proportionate risk proportionate policy. So let me talk for next few minutes on the rules for USDA SQL validity situation. Professor Pental spoke about the global. Let me spend a few minutes talking about the US rule. So US secure rule, which is basically a sustainable, ecological, consistent, uniform, and responsible, efficient. That's the full form of secure. This is the first comprehensive revision of USDA biotechnology rule, and this replaces part 340. And what is the objective of this revision? The objective is regulating organisms developed using genetic engineering for plant-based risk with greater precision. So they are going to focus on genetic engineering that lead to plant-based risk, and thereby reducing the regulatory burden for organisms that unlikely to pose plant risk. That's the basic premise. So it has got express exemption, and these are listed over here. So a plant that contains a single modification of a type in one of the following three categories are basically exempt for regulation. A change that is resulting from cellular repair of a targeted DNA break in the absence of externally provided repair template, or a targeted single base pair substitution, or introduction of a gene known to occur in the plant's gene pool, or a change in a targeted sequence to correspond to a known allele of such a gene, or to a known structural variation present in the gene pool. And that's one of the examples that I showed was an LB1. In addition, the new rule applies also says that a plant that contains a plant trait mechanism of action combines a combination that APIS has already evaluated under the previous or revised regulation and determined by not to be regulated, and only a couple of months back, last month actually, July, USD proposed for express exemptions for three additional classes, and since then the public comment period has completed, and it is expected that those would also come up as the first three. So in case a plant is not if a plant is not an express exemption, what happens? There are two regulatory paths that can also lead to a non-regulated determination. For example, the first one is exemption extension that requires a factual ground that the proposed genetic modification could be achieved through conventional plant breeding. If you are able to prove that, then that is exempt, and the timeline is 12 months. And the second one is regulatory status review, so determination of plant pest by a risk assessment of plant trait and mode of action, and expected changes in the metabolism, physiology, and development. This is a two-step tiered process, and the first step takes 1 to 180 days, and step 2 is required only if the agency requests for additional information. That too needs to be completed within 15 months. And the reason that I'm citing this example, I think this is the need of the hour, even for our regulatory agencies to have a timeline, and this I think gives a really good example of how timeline-based regulation can inspire innovation downstream. I'll not spend a lot of time on this because Professor Pental spoke about it. The couple of highlights that I would like to give here is that the Latin American countries were one of the first to accept genome editing, and this is from the recent review in vitro cellular development by Jenkins et al. They were the most progressive one, and one of the first to come with the case by case determinants, and that's why they're green. US is as you can see is also green, which are defined exemptions. Canada is unique in that it's triggered by novelty, and the countries in blue are where the discussions are ongoing. Interestingly, EU still is red, but UK has come out of that situation, and UK is going ahead. Recently, as you may have seen, that they have allowed genome-edited wheat to go downstream. Nigeria has already, among the African countries, Nigeria has already moved ahead. It's green. Ethiopia and Kenya are in the queue. South Africa, so is South Africa. India, Russia, China, three of the most populous part of the world are still debating. And as Professor Pental said, we may see something suddenly from China. There's something maybe going on. We don't know, but this is something to keep in mind. The last example that I want to show is Japan, and this is the great news, which is listed in my next slide. So these tomatoes, which are high GABA rich tomatoes, are available for sale in the market. This is a newspaper article from yesterday. These are on the shelf, and Japan is not normally considered as if you look to the earlier stage that one of the progressive country, but Japan has really embraced genome editing. With that, I'll stop and hand it over back to Professor Mishra, and I'd be happy to take questions whenever they are required. Thank you so much for the opportunity again.
Thank you. Thank you, Dr. Mohanty, for very informative presentation. And lots of questions are coming in the chat box, so I'm going to put them at the after the next talk so that we can all discuss together on various issues that the audience is raising. So our next speaker is the young scientist from IGIB, Dr. Debojyoti Chakraborty. So Devo has done phenomenal work on CRISPR Cas9 enzyme, and he has used it to really appropriate ways, both for diagnostics and also I understand he's working on clinical aspects of this, finding genome editing and using it for curing disease like sickle cell anemia, where he's made tremendous progress. And he has adopted this technology for the COVID diagnostics, which has been commercialized already. So that's a remarkable contribution in very short period of time he has done, and he's done very quality research in this area. So he is going to talk about the genome editing for the therapeutics. We have seen the agriculture component. Now we'll see the clinical aspects of this. Debo, you can share your presentation and start. You have to unmute yourself.
Can you hear me, sir?
Perfect. Yeah. Yeah.
Okay. I have a huge problem with the internet connection here, so I'm speaking through the phone and presenting on the computer. So I hope I can manage.
It's working well. Yes.
Okay. Okay. So just please bear with me in case there is any glitch. So I think you can see my screen now, right?
Yes.
Okay. Good. So it's an absolute honor to be, invited today to talk for this INSA session, and with absolutely great scientists. So it's just a bit, scared a bit to present before all of you. But thank you, sir, once again for allowing me to present. So I'm going to talk today a little bit about some of the work that we have been doing in the lab and the major goal that we have been having since I started work here in IGIB is to try and improve the CRISPR technology towards possible therapeutic applications in Indian patients. That has been the overarching goal, and in the process we have kind of done a few other things which I'll just touch upon briefly. So, just to bring everyone in the same page, it's not a science fiction anymore, and we are at this point, some people in this world who are going around with their genes edited using CRISPR. So that's where it has reached from the point of in 2012 when the first papers came in, where you could show this in a test tube, to the fact that you can now really cure diseases using genome editing. That's the level that it has reached. And some of the first diseases that have been targeted and patients are being followed up so far pretty well is, of course, sickle cell anemia, hematological disorders where possible correction of about 30 to 40% is good enough to give some kind of a advantage. And these studies have already gone, it's been there from 2019-20, and these patients are doing very well so far with no signs of the disease. So that's quite remarkable. But even more remarkable is that, now it is possible to even put CRISPR as mRNA right into the bloodstream, and that gets into the liver and corrects the disease. I think that's just tells you how powerful the technique is, and I think the previous speakers have told a lot about it. I did see Professor Thelma in the comment section talking about off-targeting, and that's really a very, very important point. And to understand why that is important is you can imagine, DNA is composed of letters, and here we are causing a break in these letters somewhere, just relying on a 20 base pair homology. Okay, so it's very important to understand that just like a PCR primer never gives just a single band in the hands of a very young PhD student, and that's because it goes and binds to other parts of the genome. Similarly, these 20 bases can actually go and bind to other parts of the genome as well with limited homology, unless you really design it so well, which comes from the point of view of the bioinformatics analysis. But it also is important to understand that the Cas enzyme itself might be also prone to binding to these places relatively strongly. And that is the concern, I think, that Professor Thelma was mentioning. And it's very important to understand what is an off-target and what is a non-target. Particularly when you see that inside a cell, and this is a video which was taken using atomic force microscopy. I hope it plays though. Oh, here. Okay, so by some colleagues in Japan who later on started working with us, I think it's not playing probably, but let's see. Yeah, so I'll just push it along. So as you can see, this is the Cas9 protein, which is sitting on a double-stranded DNA. And if you look at, how long it takes for the DNA to cut, it is just a few seconds. And that's how rapid this entire enzymatic reaction is. That within a few seconds, a single molecule of protein sitting on its target is able to make this break. And when you think about it, that's how fast the situation is inside the cell. And you have targets and you have off targets, which can also be bound by Cas. Then, regardless of how much time the Cas is really sitting inside the nucleus, I know there are a lot of improvements made in terms of delivery and reducing the time taken for editing. This is actually in seconds how long the Cas takes to cut the DNA. And this is what we observe also in the lab. Now, the problem of this is that if it is at the target site, then this can be repaired and you can change things there and so on. But when it happens at off-target sites, which can be sometimes really kilobases away from that intended target, you can have large chromosomal changes, and these could be alterations such as truncations and translocation events and so on. So these could be different types of things that actually happen. And this is really the concern as far as the effects of nuclease-mediated targeting of CRISPR and so on. Now, coming from there, you really need what you is something which is highly specific, okay? And the specificity should come at the level of the CRISPR system itself. And what people have been doing is that ever since they started working on the Cas9 from Streptococcus pyogenes, which almost 99% of the labs across the world use, they have been trying to improve this using protein engineering, protein evolution, and so on, into versions that are much more specific on the DNA. That is, they recognize only the target, but they do not recognize, let's say, or cut the off target. And that's why what you are seeing over here is that these additional, mutations or additional changes on the Cas9 led to the discovery of things like sniper Cas9, ESP-Cas9, high fidelity ones, and even evolved versions, which were evolved in the Cas9 milieu, basically. But one important thing to understand is that generally, when you take a highly active Cas system and you start improving it such that its off-target contacts become lessened, they actually reduce the general activity as well. So the specificity increases, the general activity decreases, and that's a two-edged sword when you try to make changes in the protein. And nevertheless, you can still have pretty good balance between specificity and activity, and these are some of the cases which have also kind of gone towards clinical trials, and people are pretty happy with the way that that they are working in terms of specificity, but with reduced activity. I would like to keep this, to keep these two cases in mind because they will come up later in the talk as well, ESP-Cas9 and high fidelity ones. So now what have we been doing? In 2019, we actually published a paper, and all this work that I'm going to show you today is with close collaboration with my colleague, Dr. Shobhik Maiti, who brings in all his biophysical expertise to study these problems. And this particular study, we showed that we extensively characterized an orthogonal Cas system from a bacteria called Francisella novicida. It was very poorly studied, principally because it was a big protein, and people have always been looking for smaller cases which can be packaged in vectors, which like AAVs and so on. This Cas was relatively big, but very interestingly, because of the completely different way evolutionarily that it has it has evolved, what we saw was that the contacts that it made with the DNA bases in the region where it interacted with the DNA was very different from the structure of the other Cas2 class of Cases. And that made it highly specific. And specificity here means whether it could be able to tolerate mismatches on the target. So if you take a target with mismatches such as just 2 nucleotides or 3 nucleotides, your standard Streptococcus pyogenes Cas9 would still cleave them. And this is inside a test tube. You can imagine what happens inside the cell. But we found that this wild type FN Cas9 was not able to cleave these targets, which made it really an interesting kind of a discovery that it was very specific. Now, that was also the same time that CSIR launched its sickle cell anemia project, and a lot of labs became involved in it. It's a very dangerous disease, and US is of course launching these clinical trials for CRISPR therapy in US, but India is no far away as far as the patients are concerned. It's unfortunately a disease which is present mostly in the tribal belts, people with very low access to healthcare and so on, and it's a debilitating disease, as you can see over here. Some estimates say that about more than 50% of the carriers of the world actually come from India. And it's a single mismatch, single point mutation, which causes glutamic acid to change into valine. So it's a very ideal case of correcting to completely cure a disease. If you could potentially correct this mutation, you could potentially reverse the disease completely. Now, what we found out was that if you use this FN-Cas9, which we were characterizing, and you use it at the same locus where this mutation is, we could edit it, but unfortunately the editing was very low. If you look at how much SP Streptococcus pyogenes Cas9 was editing, this was way lower than that, and that would not be enough if you would want to therapeutically give some benefit to the patient. This meant that if you had to do something like this in a patient, we would need, but of course the Cas9 was very specific, so we didn't want to let go of it either. So if you would need to use it for a patient cure, we would need two things. One, we would need a highly efficient variant which can increase the editing percentages, but importantly, it would also be able to retain its on target specificity, which is this USP of this Cas system. So to cut a long story short, we collaborated with one of the pioneers of Cas9 structural biology who solved the crystal structure of Cas9 in the first place, which is Osamu Nureki at the University of Tokyo. And we made a bunch of variants of Cas9, close to 50, 60 different versions of FN-Cas9 with all site-directed mutagenesis and so on. And it's okay to say about it today. We have filed a patent from CSIR on this aspect as well. But what we actually figured out was that through this protein engineering and through the site-directed changes, you can really increase the kinetic cleavage of the Cas9 from a wild-type version, which is here, to something which we are now calling as enhanced FN-Cas9 or ENFN-Cas9. These are certain variants. There are several of them, but these are our favorite ones, which are which will be later on called as EN1, EN15, and EN31. So these are they really, at a very short period of time, the Cas can cleave the target very fast. Now the question was, if it's cleaving so fast, what does it mean? Does it mean that it is now just able to recognize DNA faster, or is it binding to DNA stronger and thereby the kinetic activities have increased? So it turns out that if you do very, very careful binding experiments of target DNA with Cas, you actually see that because of this engineering, these engineered Cas proteins are now able to bind to DNA almost 3 times stronger than the wild type FN-Cas9. And this is now the same binding affinity that a wild type Streptococcus pyogenes Cas9 actually binds to DNA with. So we also did those experiments. So the engineering is actually happening because there is now stronger DNA protein interaction. Now, these two aspects, that there is a specificity and then there is a stronger DNA binding, was something that we made very good use of in a kind of a side project, which I think some of you might well be aware of, which is the Feluda technology, which was the CRISPR DX platform, which started from CSIR, went to Tata, and now there's a product which is approved, got a FDA, got a DCGI clearance, and now an EU clearance as well, CE approval as well, and going into different places. But it essentially uses the fact that the Cas system can recognize anything with very high affinity and will not recognize anything even if there are mismatches. And that is the principle of this FN-Cas9 editor link uniform detection assay. And we combine this Cas biology with paper strip chemistry so that if you have a disease, even with a single point mutation, which such as sickle cell disease, you can actually detect that on a paper strip. And that's how the resolution of this of the detection basically. And with this in engineered versions, you can improve the detection as well on a paper strip. And the idea was that normally if you have to go looking for mutations, you would have to sequence them. Can we do this without the need for any sequencing at all? That was the whole thing. And we also developed 2 other things, because there are Bengalis in the group, and there was a Feluda, so there also was a Topshe and a Jatayu. So Topshe is the app which is basically taking a picture of the strip, and it's machine language trained, so you do not need to rely on your eyes, so you can get the result, whether it's a COVID positive or a COVID negative. So that's true outcome predicted via strip evaluation is Topshe. And then there is a web server called Jatayu, which is a junction for the analysis of your Feluda experiment, where you put in your disease of interest and you get the Feluda primers designed for that. Of course, this has now, like I said, has been commercialized, and it's Tata MD has taken it into a robotic platform through a kind of how to say automated system, and they are going to different places where normal RT-PCR testing has not been able to reach, and so on. So those are things that that have been happening at the CRISPR DX platform. But let me come back to the main story, which is the engineering part. So what we did then was to see that there were some reports which said that if you change amino acids in the protein backbone, which is responsible for protospacer adjacent motif contacts, which is called the PAM. And I think if some of you are in the CRISPR field, you will know that the first sequence that the Cas interacts with on the DNA is actually something called a protospacer adjacent motif, or a PAM. If you have that PAM, then the Cas is able to open up the DNA, and then the guide RNA can go in and start pairing, and then it changes conformation and so on. But if you do not have that PAM, then if there is a disease mutation, then you cannot access it. Now, the FN-Cas9 was actually a NGG PAM, or a triplet nucleotide, which is just like the SP-Cas9. But what we found out was that after engineering, this has now become an NRG-NGR PAM. So that's basically NGA-NAG PAM. So that automatically increases the total targeting range in the entire genome to almost twice or thrice of the wild type FN-Cas9. So means more disease mutations can be targeted, more mutations that can be diagnosed, and so on. And that makes it actually just the number 3 at the moment among all cases that have been made or engineered so far. This is just a snapshot of that. How we found out, we did through this experiment. I won't go into details, where we took a bunch of plasmids with all possible combinations of 8 nucleotides, and then cleaved them with the Cas, then did a dropout and did a DNA library preparation, next generation sequencing to see which are the PAMs that were recognized. Now, when you can recognize more PAMs, the advantage of that is that you can now recognize more mutations, which you would not be able to do before with, let's say, an NGG. And one of the places where we found a very good use of this is actually the variants of concern in SARS-CoV-2. So this is a way by which we could, on a paper strip, detect if one has an alpha variant or a delta variant or whatever it is. And that simply happens because now the Cas9 can recognize more protospacer adjacent motifs in the genome. So the FN-Cas9, being highly specific, will not cleave or bind to a substrate where there is a single mismatch, but will cleave or make a contact when there is a when it is completely matched, basically. And that can be used for a readout, such as in a paper strip. So we could now essentially cover all the mutations that are there as a signature mutations for all the variants of concerns that has been seen in the SARS-CoV evolution. So whatever you are doing through sequencing, you could potentially not require the sequencer as a signature variant detection. You can, of course, we need sequencing to know what are the mutations that are arising and how it is evolving. But if you simply have a sample and you want to know if it's a delta variant or not, one can do this kind of a thing. So this is Ray. That's again no questions asked why the naming. So that's rapid variant assay, and this is something that we are currently negotiating with World Health Organization to see how this can be deployed. Of course, the other advantage of it's another snapshot, is that you could use this now to cover more mutations for therapeutic correction. And this is an example. He's a Mayank Bansal. He's a ophthalmologist, MD. He's working towards a PhD in our group. And he came up with a patient who had an Usher syndrome mutation where there was no PAM nearby. So technically, he could not be corrected with the wild type version of the Cas. But the engineering, of course, now allows us to target something which is causing the disease, a C2T, and has an NGA PAM. So, the direction in which we go, we made the iPSCs, we are now correcting it. And I think with our very, hopefully, if our collaborators I think I saw Dr. Orko was there in the panel. If these things go ahead, then we can actually be able to look at possible AAV-based therapies where you could package these as base editors and deliver. So now coming back to, we made these engineered versions, and I again come back to ESP, SP-Cas9, and HF1, which is like a kind of a standard for good specificity and activity. When we put this into cells, what we see is that these engineered FN-Cas9 actually behaves better than even these high fidelity Streptococcus pyogenes variants. So that it means that it's much more efficient. But more importantly, even at validated GuideSeq or basically off-target sites which have been validated by sequencing, you do not see any speck of editing anymore. So that makes them retain that high specificity that they have. Not only that, I mean, when we design these off-target assays and so on, we generally have become better in developing guide RNA. So we typically do not target off-targets with, let's say, 2 mismatches or 3 mismatches. We do more. But if you actually have an off-target with a single mismatch, then it appears that these high fidelity SP-Cas9 is unable to really discriminate. So they tend to cleave these off-targets with even a single mismatch pretty well. So these are things that we're appreciating more and more. ENFN-Cas9, due to a very, very different way of negotiating with DNA, is still maintaining its high discrimination. So that's where it leaves the wild type. This is where it leads this single mismatch off target, which is very, very low. When you put it inside a cell as a therapy, of course, it goes even lower. But that's not the case with some of the more high fidelity versions. And when you cleave the target more, you cleave the target more efficiently. You can also do very nice genome editing. So this was actually a 4 kb construct which we entered, which we inserted into the genome at a very specific site, basically. And we see that it can very nicely integrate way higher than any of the other Cas9s, even the ones which are have fidelity versions. So all this is good. It's very good to get paper and do all this in the lab. But the major aim that we always had was to see how we can take this to people. That's one part of the puzzle, to develop good editors, precise editors. The next part of the puzzle is to actually deliver them. How do we put them inside patients? We have done these experiments in patient cells also, hematopoietic stem cells, and we see that even with a wild type FN-Cas9, we can correct that sickle cell mutation pretty well. It's about 8%. But like I told you, that we need about 20% and 30%, which is where the engineered FN-Cas9 editors come in. But even if we have them, we would need to deliver them. And for that, we have started, working with people who can help us this. So for adenovectors, we are collaborating closely with Andre Lieber at University of Washington, where we have packaged the FN-Cas9 into adenovectors, testing them, because adenovectors, I think they have been in trials for a long period of time. Covishield and so on everybody knows about these vectors now and so on. Of course, adeno-associated vectors have their own advantages, but it requires a smaller sized Cas9, because adeno-associated vectors are they have a shorter packaging size, but they also have some advantages for certain disorders. And for that, what we did is we again engineered the Cas9 and made it into a shorter Cas9. So if you look at this domain over here, which doesn't make any contacts with the DNA, we deleted this and we call it now as the mini Cas9, mini ANFN Cas9, which has the same efficiency and same specificity as the Cas9. So like I said, if all goes well and if we can work together with Orco, we can see how this can be then later on taken for some diseases. And of course, at the CSIR level itself, we have been trying to see with IICT if we can deliver them as RNA itself. I think the RNA viruses has just the RNA vaccines have just increased the scope and applicability of such delivery vectors. But that's the last slide. I won't go into more. Very important to understand that this is all good in theory, unless we can actually do it in the patient. That's the real challenge. And it's very important to know that what are the challenges. One of the important things is the genome fidelity. When you put these into the genome and you let them be there, what are the things that are doing at the genomic level? Does it really cause breaks, and till what extent? The other thing is, of course, immune response to the vector, to the Cas9, etc. There are antibodies which are there which recognizes the vector. How does that impact? And is it something that you can still work with? What is the correction, efficiency of in vivo correction? But more importantly, what is the cost? We are talking here of a disease, even if it's sickle cell, which is a tribal's disease. It's a disease by people who are really needing healthcare even for the basic amenities. If you have to bring genome editing to them, you have to also think about more frugal innovations, which are things that need to be at the cornerstone for any kind of such wonderful technological advance. So I think those are the things that we also are having in mind, but I think it's a common thing that everybody should be more discussing about, I feel, to bring down costs and where to, at what level. And if all this goes well, we are very excited to go towards the phase 1. At the moment, we're going into preclinical now, very soon, hopefully after the preclinical results are out. So with that, this is probably the most important slide. So a lot of people to thank for, and I think we're generally very well funded. It's a big group working on multiple things. So yeah, I would be happy to take any questions that you might have.
Okay, thank you, Dova, for very nice work and very pertinent demonstration of use of quality science for applications. So we
Thank you.
Reached up to diagnostic. Hopefully, it will reach clinics as well sooner or later. And yeah, it gives a lot of hope that quality science is being done, which is coming closer to the applications. So with that, there are lots of questions that have been coming in the chat box, and so maybe Dr. Nautiyal, we can have few more minutes because we are, although a little bit late, so we can cover some few questions.
Could you be reading them out?
Yeah, I can read them out. So some are simply the comments, but I can share with others who are not reading the chat box. Uh, Dr. Bhaskar Balakrishnan mentioned that we must increase our research budget from 0.7% of GDP to 3%. UNESCO has benchmark of 2%. I think we all agree with that. And more number of people should come to research, better talent should be attracted. Is absolutely necessary if we want to really become a powerful country, that we have all the potential. Dr. Bansal mentioned that there should be an inter-academy working group that needs to be developed to highlight the issues in order to promote the use of these advanced technologies. He was talking after Dr. Pental's presentation, which is appropriate, and I'm sure INSA will consider this. I think we have to, considering what science has delivered in fighting against this pandemic, and as Devo mentioned, vaccines have really made the way, and science is more and more appreciated by people. Maybe academies should use this opportunity and convince various stakeholders in adopting these technologies. So there are a few questions to Dr. Mohanty. Dr. T.R. Sharma is asking how editing of vaccine gene of maize also help in increasing the yield of maize. So Dr. Mohanty would like to mention in maybe very briefly.
Yes. So what happens is that, as I mentioned, that this particular mutant allele is a very old mutation, so that means that is in a non-elite background. So when you try to bring that mutation from non-elite to elite, in spite of repeated round of backcrossing, there is still chunk of genome that is coming from your the original allele, which is non-elite. That leads to reduction of that about 5.5 bushel yield. Whereas in genome editing, since you are editing the elite line itself, you do not see that yield drag. So which was their limitation. Thank you.
All right. Thank you. Then there's a question from Professor B.K. Telma, which they were partly covered. And that question was for Dr. Mohanty. Would you like to say a few words on the off-target effects of these powerful editing tools? So although they were partly addressed, for the agriculture aspect, Dr. Mohanty, you may want to just make a very brief comment.
Yeah, that's an important consideration. And what we have done is we have done evaluation of these. So basically, with a good designing guide, you can take care of the problem. And there is a paper that I did send it to Professor Thelma that was published by our group in Scientific Report, and the title is CRISPR-Cas9 editing in maize: systemic evaluation of off-target activity and its relevance in crop improvement. So what we found, we designed targets and evaluated them downstream in transgenic in the genome-edited corn lines, And what we found was that with good design, we could take care of most of the off-target effects. And there was a rule that we came up with, if we have 3 mismatches, including one in the PAM protospacer region, you should be able to take care of most of the mismatch. And this paper, please do refer, because we are we have showed there that the number of off target or mismatch that we find is much less than the normal variation that you see in plants. So please do refer to this paper, which has got a lot of details. Thank you.
Thank you. Now there are few questions. I will not be able to take all the questions, but few now to Debo. So with the engineered variant of Cas9, now PAM is not a limitation and we can edit any sequence. Would you like to comment, Debo?
So we can edit any sequence with a PAM containing NGR and NRG. So that means that instead of NGG, it's going to be NGA and NAG. So those can also be edited. It is still not PAM flex, which means that it can go anywhere. But I think that typically covers almost 70-80% of the genome, basically.
All right. So the second one is the related question by Avnish. How did the PAM site of FNCAS9 change from NGG to NGN?
I do not know the answer to how, but I can tell you, how we did it is by engineering PAM duplex mutation, contacts. So we changed it to amino acids that are now able to make hydrogen bonds with residues where it was previously not able to do. So I mean, we're putting this up on a server, so you'll be able to see more details of how this engineering was done.
Right. So again, can we try using the wild type for our infant Cas9 in plant genome editing?
Absolutely. So these, plasmids are now freely available. We'll put them in AdGene in next week itself, probably. And also for all the people here, you can just write to me. I think at NABI Mohali, where Amitabh, I forgot his name. Who's working on CRISPR genetic engineering in bananas. He's using these engineered versions now to do vitamin A fortification in bananas. Yeah.
Siddharth Tiwari.
Siddharth Tiwari, yeah.
So one last question for you, Debo. I see if you can answer this. So how do we see genome editing in case of structural defects in the humans like congenital heart disease?
So I think the issue is delivery vectors. So if you have a mutation which causes this, so if by the name as it sounds, it's a congenital disease. So if there are single mutations that can be targeted, which can give some kind of a relief, and it can be targeted safely. And again, I think we have a very experienced delivery guy in the audience, which is Shubhra Ghosh. If there are serotypes which can specifically go to these kind of cells, then yes, there are chances. But I think just like CRISPR has boomed over the last 5-6 years, the next 4-5 years would be the same thing for delivery vectors. I think we know how to make it work in the lab, but how to make it work in patients is where the delivery vectors becomes very important.
Very true. Yeah. Yes. Another challenge is the next question for you is have you had a chance to assess the role of genetic background in editing efficiency of your patient pool?
Excellent question. I could almost imagine ma'am to ask this. No. So since we have only worked with limited, let's say, limited patients so far. I mean, of course, we are going towards a bigger, but there the variation has been not studied to depth using sequencing. So these are sickle cell patients where the whole genome sequencing has not yet been done. But I think CCMB has done fantastic work with Dr. Rakesh's involvement during the sickle cell project, where they have done a lot of whole genome sequencing for sickle cell patients. So yes, at some point we can take all these two things together and see how editing is perceived in the background of mutations.
Thank you, though.
Thank you.
So if I can ask one question, which is little general kind to Professor Pental. How you think, I mean, you have been handling various agencies and your voice matters. And I think it's very important time, both in terms of that we need these technologies, both for the healthcare and for the agriculture. At the same time, I see it as an opportunity that there is a more trust in science. I mean, less than a year, vaccine was never ever, I don't think anybody has imagined that it will happen. And they're so successful. So can we make use of this positive trend, if I can say, and package these things as a scientific community to take these technologies closer to societal applications. How would you guide us, and what advice that we should put together as a one front?
It's there is an opportunity. I agree with you, because who would have heard of virus mutating? Who would have heard of virus creating such a havoc in terms of a pathogen? Really, I mean, this is a good example to show how arms race goes on between pathogens and hosts. And in plants, you see the things are easier because of even off target. Mohanty has mentioned some developments and better type proteins or more specific proteins will be useful. But we mostly do agrobacterium based introduction of this CRISPR-Cas into the into the plant cells, and we have to backcross it to segregate out the tDNA so that there is no foreign DNA left in the plant. So during backcross breeding, 2-3 backcrosses, the original genome will be restored, even if there are minor changes which are off target. So that is why many countries in the world are really feeling very secure about SDN1 and SDN2 type of interventions. But I agree with you. I think this is a good opportunity to sensitize the policymakers and the government on need for additional money and the need for using the new technologies. I think we need to gain trust into and the best placed are our academies. They're doing excellent work in terms of so many initiatives have been taken by our academies in putting young students into labs, scholarships, and so on and so forth. But I think this is one area, science, to showcase science, to make it how relevant it is for the society, I think is something which we should all contribute to. It's not only the presidents can do it all the fellows will also have to contribute, only then it will go forward. But I think it's a good opportune time to do this.
Okay. Thank you, Professor Pental, for your comments. I will now hand over to Dr. Nautiyal for the next step. And I would like, before that, thank all of you for participating. And I think this is something which we should take it as a initiation, and I'm sure INSA and SIR will continue to pursue the issues that are being raised because they're so important. They cannot be left unattended. And I think it's our responsibility that we see that these are in the front page till we are able to convince, because these are futures. If we don't do it, we will be far behind and keep buying things, be ruled by others economically. So with that, I thank all of you and I hand over to Dr. Nautiyal for the vote of thanks. Thank you.
Thank you, Dr. Rakesh Mishra, for conducting such a lively discussion. And of course, our experts were outstanding people. The science fiction of yesterday is turning into reality faster than anybody could imagine. Thank you, Professor Shaha and Dr. Mishra, for conceptualizing and conducting such a beautiful discussion on such a relevant topic, and also thinking of this exciting topic for discussion and creating this looking glass platform for the whole scientific community. Professor Deepak Pental, a veteran and a very well-known authority in the field, Dr. Amitabh, Mohanty, and Dr. Debojyoti Chakraborty, a big thank you to all sirs for this wonderful contribution to the science of the country and for turning this idea into a reality, which Professor Shaha and Dr. Mishra had conceived. The support from the Science and Engineering Research Board is thankfully acknowledged. We also acknowledge the support by INSA staff, including Dr. Arora, Dr. Zukarkar, Dr. Bhattacharya, Dr. Mandal, and especially Mr. Karthik, who has taken care of this platform. So at the end, reminding you that we are meeting again on 23rd of September during 3 to 5 p.m. And again on 28th September from 3 to 5 p.m. for a program which is being conducted by the Kolkata chapter of INSA. And of course, my vote of thanks will be incomplete without thanking those wonderful people who asked such good questions and made this really an interactive session. So again, with thanks to all the panelists and to all those who contributed to this program. Thank you so much. Have a safe.
Thank you.
Okay, thank you. Bye-bye. Bye, everybody.
Thank you. Thank you.
And thanks, Dr. Mohanty. We kept you awake for odd hours.
Thank you for the invite.
She was actually anyway.
Thank you, sir. Bye-bye.